Good afternoon. My name is Nancy Sloan,
I'm Quinn Nystrom.
And I'm Earl Gilbert.
and our research is on the visual cortex and the effects of the visual cortex lesions on perceptual grouping in rats. The visual cortex is the part of the brain that processes visual information. There are two pathways that this information can travel down. One goes from the eye to the superior colliculus to the lateral posterior nucleus and onto the medial and lateral prestriate cortexes. The other goes from the eye to the dorsal lateral geniculate nucleus to the striate cortex. The way our brain processes information is through three general steps. First, the brain receives the visual stimulus. Next, an intermediate step of processing the information occurs, followed by higher processing such as pattern discrimination, or being able to pick out different solid patterns. A process called grouping, in which the brain organizes individual elements into recognizable patterns, is a part of the intermediate step of processing. The big question we asked ourselves is whether or not grouping and pattern discrimination are related. We hypothesize that grouping is a necessary precursor to pattern discrimination, and thus the two are related. Therefore, by creating lesions in the brains of rats, both grouping and pattern discrimination should be affected post-surgery. The goals for our experiment are to first identify the grouping areas, or the cortical areas involved in grouping, and second, to determine whether these areas involved in grouping are distinct from the areas in pattern discrimination. In our experiment, we used a customized operant conditioning chamber, which houses a small chamber and glass funnel for the rat's head, bordered by a water funnel on either side of the chamber. The rat places its head in the chamber, and after a half-second delay, a visual stimulus is displayed, and the rat will go to one of the water funnels to receive water after correctly recognizing the stimulus as a group of horizontal or vertical lines. If the rat is incorrect, the light in the chamber goes out for 4 seconds. In order to prepare for the experiment, 19 rats were deprived of water 22 hours leading up to the experiment, and were trained by displaying 4 horizontal or vertical solid bars, and to pass on to the trial stage, the rats had to get 90% of the 40 sessions correct. In diagram A, it shows the top view of the chamber, and in diagram B, it shows the top view of the head position, and the solid line represents the region that is viewed through both eyes, and the dashed line represents the visual region through one eye. So we used relative proximity to distinguish between more or less recognizable groups. The distance B remained constant between blocks, while distance A became smaller in a certain direction, creating the appearance of vertical or horizontal lines. Therefore, the higher the relative proximity, the easier it is to group the blocks into a line. So we split the rats into 5 different groups. Group A had lesions in area 18. There were 3 rats in Group B that had lesions in area 17. There were 5 rats that had lesions in area 18A, and there were 3 rats that had lesions in area 18A lateral. And there's also a control group of 4 rats that were anesthetized, yet no lesion was taken out. We also gave the rats 1 week to recover from the surgery before performing tests.
So before we begin discussing the results of the effects of lesions on grouping and pattern discrimination, it is important to note the effect on reaction times. In early training, the responses were spread over a wide variety of reaction times, while after training, majority of the responses fell under very short reaction times, most under 150 milliseconds, as seen in Figure B. This shows that the reaction time decreased as the rats learned how to respond to the stimulus. Figure C shows the range of reaction times when there is no stimulus. This will act as a control in order to compare the reaction times after the surgery is done. If the reaction times post-surgery are the same as the reaction times with no stimulus, it would indicate that the rat is unable to perceive any stimulus at all, and the surgery affects more than just the ability to process grouping or pattern distinction. However, as seen in Figure D, the reaction times emulate the reaction times in Figure B after training. We can assume the surgery did not affect the rat's ability to receive stimuli as they were able to react quickly. So now let's move on discussing the results of the solid line discrimination test. Before surgery and after training, the rats were over 90% correct in their discrimination of solid lines. After surgery in the control group, the group that received the sham surgery, the group that received ablations in area 17 and lateral ablations in 18A, all were able to maintain their accuracy of 90% correctness. However, groups that received lesions in area 17 and 18A non-lateral dropped in correctness significantly, showing below 60% correctness overall. Finally, we will look at the dot discrimination results. Before surgery, we see that as relative proximity increases, the percent correctness increases in all groups. This is because, as seen in an earlier figure, that the grouping becomes more obvious as the spacing between the dots decreases. This trend stays consistent in the control group, and the groups within lesions to areas 18 and 18A lateral, seen in figures A, C, and E, respectively. Figures B and D show that correctness in dot discrimination decreased significantly after lesions were applied to areas 17 and 18A.
Grouping is an early component of visual processing. As seen in the experiment when areas 17 and 18A are disrupted, both pattern and solid line discrimination are greatly reduced. This shows a positive correlation between the grouping function of the brain and higher-level visual functions. Correlation, however, is not causation. The results gathered from the experiment could also be affected by other regions of the brain, specifically areas 35 and 36, which are involved in higher-level visual processing. In Figure A of the visual here, you see an example of the stimuli presented to the rats while they were in the customized operant conditioning chamber. While viewing this stimulus, certain parts of the rat's brain activated, as shown in Figure B. Previous versions of this study allowed test animals to move around in the chamber while visual stimulus was presented, creating inconsistent distance and angle to the stimulus. In addition to the variability of input, previous studies also provided post-operation training for the animals, allowing for slight recovery of perceptual ability. These inconsistencies in the experiments skewed the final data gathered. Despite improvements made from previous studies, this experiment still has its flaws. The lesions used in this experiment were not confined to their assigned areas, instead running over into neighboring areas and affecting the final data gathered by the experiment. The experiment also didn't rule out the effects of other regions of the brain, which might affect the results. Other additions to the experiment include perception of colors or multiple inputs and discriminations.
There are a few possible applications for this information that we've learned. First, it just increases the knowledge and understanding of mechanisms within the visual cortex and shows the relation between pattern discrimination and grouping. Similar to the idea that we learned about of viral neuronal tracing, being able to relate an area to the brain and these functions could tell us what part of the brain is affected if an organism has problems with relation to these brain processes. So this expands on previous knowledge about pattern discrimination. It has been studied before how lesions to the visual cortex in general will affect pattern discrimination in rats. However, this experiment expanded on what specific areas will have different effects on both pattern discrimination and grouping, so it makes the knowledge that we have more specific and more applicable. This concludes our experiment on